Welcome to this explainer. Right now, at this very second, inside your bloodstream, there is a microscopic, highly violent, and incredibly coordinated special ops team out on patrol. When we think of immunity, we usually just picture white blood cells, or maybe antibodies, right? But there is this whole other hidden, underlying network that is absolutely vital to your survival.
It really begs a fascinating question. How does your body literally punch holes in invading bacteria? Well, to answer that, we're going to have to uncover a lethal biological cascade that acts as your body's ultimate, highly specialized defense force. It's a system so powerful that it operates completely under the radar, yet it can obliterate a pathogen in literal fractions of a second. So, here is our roadmap for today. First, we'll meet your body's special ops. Second, we'll cover the three tripwires and their shared target. Third, the membrane attack complex. Fourth, preventing friendly fire. And finally, number 5, we'll look at what happens when the system breaks down.
All right, part 1, let's meet your body's special ops.
First off, the sheer scale of this system is just incredible. The complement system isn't just one single thing. It's actually a collection of over 30 different plasma and membrane-bound proteins, and they're synthesized mainly right there in your liver. They constantly patrol your bloodstream in a resting state as inactive zymogens. Basically, think of them as these sleeping soldiers walking around with their safeties on, just waiting to be triggered. Now, their name, complement, historically comes from this old idea that they just assist or complement your antibodies in clearing out microbes. But as we're going to see, they are incredibly formidable all on their own.
So what do they actually do? Well, this team has 5 major missions. First is opsonization, which is effectively putting a glowing neon target on a pathogen or, seasoning it with hot sauce so your immune cells really want to eat it. Second is lysis, which is the actual physical perforation and destruction of the target's membrane. Third, they drive inflammation and chemoattraction, literally recruiting heavy immune backup to the site of an infection. Fourth, they clear out old immune complexes, basically acting as the cellular garbage disposal. And finally, they regulate your overall immune response, acting as this critical bridge between your rapid-fire innate immunity and your highly specific adaptive immunity. It's just an incredibly versatile toolkit.
Moving on to part 2, 3 tripwires, 1 shared target.
Okay, so how does this dormant army know when to actually strike? There are 3 distinct alarms that can wake up the system. The first alarm is called the classical pathway. This one trips when specific antibodies like IgM or IgG have already tagged an invader. A complement protein called C1 comes along and binds right to the tail end of those antibodies. And the moment C1 binds, boom, it becomes active and starts this rapid protein cleavage cascade, basically chopping up downstream proteins to build a specialized enzyme. It's essentially your adaptive immune system explicitly calling in an airstrike from the innate system.
Then we had the second alarm, the lectin pathway. And this one brilliantly illustrates just how resourceful your body really is, because this pathway acts completely independent of antibodies. Instead, it uses sensors like mannose binding lectin to directly spot specific carbohydrate or sugar patterns right there on the surfaces of microbes. Once these sensors latch onto those foreign sugars, associated enzymes kick in, and they start chopping up the exact same downstream proteins as the classical pathway. It's a direct, innate recognition system. It doesn't even bother waiting for antibodies to be produced.
And finally, the third alarm is the alternative pathway. Now this one is super unique. It acts as a constant, spontaneous patrol. Right now, in your plasma, there's this low-level spontaneous activation, often called tickover, of a protein called C3. This means the system is literally always idling, constantly testing surfaces it bumps into. If this idling system bumps into a hostile microbial surface, it binds with other factors to create a really powerful enzyme. But it's more than just a standalone trigger. This alternative pathway acts as a massive background amplification loop. It basically takes the signals from the first 2 pathways and cranks the volume all the way up to 11.
Now, what's truly brilliant about this biological design is its convergence. No matter which of those 3 tripwires gets crossed, classical, lectin, or alternative, all roads lead precisely to the activation of C3. Once C3 gets cleaved, it cascades right down to cleave C5, and that specific step initiates the terminal pathway. This is it. This is the singular, explosive response that all 3 initial alarms were building toward.
Which brings us to part 3, the membrane attack complex.
Okay, so once that C5 protein is split into two pieces, the system deploys its absolute ultimate weapon, the membrane attack complex, or the MAC. This is the physiological equivalent of a heavy battering ram, and let me tell you, it is especially devastating against gram-negative bacteria and other vulnerable cellular targets. Let's actually zoom in and look at how this weapon gets built right on the target's surface. The larger fragment from our previous step, C5b, acts like an anchor. It binds to C6, then C7, and then C8. This entire complex embeds itself right into the outer membrane of the pathogen. But, it doesn't stop there. This structure then recruits multiple molecules of C9, sometimes a dozen or more of them. They arrange themselves into a literal ring or cylinder, and they punch a massive pore straight through the target's cell wall. Because of this gaping hole, water and ions just rush into the bacteria, causing this lethal osmotic imbalance. The pathogen swells up and bursts like an overfilled water balloon. It is completely obliterated.
But wait, the attack isn't just about blowing a hole in the membrane. Remember how earlier those proteins were cleaved, chopped in half to build this weapon? Well, the smaller splintered fragments left behind are known as anaphylactoxins. Think of them as chemical flares. They make your blood vessels leaky so immune backup can arrive faster, they stimulate histamine release, and they lay down this chemical trail that aggressively recruits other white blood cells straight to the battleground. The end result? Massive localized inflammation to wipe out absolutely any remaining threat.
All right, part 4, preventing friendly fire.
Because this system is so insanely destructive, there have to be incredibly strict rules of engagement. Otherwise, our defenders might accidentally attack our own tissues. So while pathogens suffer rapid destruction, our own host cells remain totally unharmed. Why is that? Well, our host cells are equipped with highly specialized molecular shields, membrane-bound regulators and plasma inhibitors, that pathogens simply do not have.
Let's take a look at some of these biological emergency brakes. In your plasma, you've got inhibitors that keep the activation pathways from spiraling completely out of control. But even more amazing are the regulators right on your cells themselves. You have proteins like DAF, which literally tear apart the enemy enzymes before they can even fire. And then you have protectin, the ultimate safeguard, which actively blocks that lethal pore from ever forming. Your cells are literally disarming the weapons aimed right at them.
And finally, part 5, when the system breaks down.
So what actually happens when you're missing one of these crucial pieces? Well, missing just one of these specific proteins translates directly to severe, real-world, life-threatening diseases. For example, if you lack C3, that main convergence point we talked about, you face severe, recurrent bacterial infections. Missing the early components predisposes you to autoimmune disorders like lupus. And if you lose those crucial emergency shields on your cells, it causes conditions where your complement system literally turns around, attacks, and destroys your own red blood cells.
And it's not just about genetic deficiencies. Even in a structurally normal system, if complement becomes hyperactive or chronically dysregulated, it becomes a massive liability. Overactivation of this system is directly implicated in devastating chronic conditions. It drives tissue damage and kidney diseases, it actually worsens the injury when blood flow returns to tissues after a heart attack, and the chronic inflammation it causes is even linked to the progression of Alzheimer's disease.
Which brings us to a final provocative thought to leave you with. We've seen how the complement system operates as this brilliantly designed hyperlethal defense force that protects us from microscopic threats every single day. But we've also seen how easily it can turn its incredible weaponry against us. So as we wrap up this explainer, I'll leave you to ponder this. As medical science continues to advance, how might future breakthroughs allow us to successfully tame, modulate, and retrain our body's most lethal special ops team? Thank you so much for joining me on this deep dive into how the complement system works. Keep wondering and keep learning.